Hello everyone, here we are going to discuss about carcinogenesis. Actually, carcinogenesis means a multi-step process. There are multi-steps which involves in carcinogenesis. It occurs due to multiple genetic alterations. There are multiple genetic alterations, not one genetic alteration. In each step or in each process, there may be genetic alteration. So we call it as multiple genetic alterations. Here you can see here is a normal cell cycle which has 4 or 5 phases. Here G1 phase or pre-synthetic growth phase. Here is S phase or DNA synthetic phase. Here is G2 phase or pre-mitotic growth phase. Here is M phase or mitotic phase, and there is G0 phase or quiescent cell phase. That means when a cell undergo G0 phase, that cell become dormant cell or it goes out from the cell cycle. It stops its proliferation. When each cycle undergo this process or normal cell cycle, each cell has to undergo or each cell has to pass several checkpoints. Here there are G1 checkpoint, G2 checkpoint as well as metaphase checkpoints. We discuss only G1 checkpoint and G2 checkpoint here. Why those cells undergo these checkpoints is in those checkpoints they detect if there is any abnormality in those cells that undergo in the cell cycle. As an example, during this G1 checkpoint, there are several molecules to recognize the abnormalities between the cells. They check for nutrients in the cell, growth factors in the cell, or any DNA damage in the cell, or other favorable conditions for cell division or unfavorable conditions for cell division. Like that, those conditions are detected at the G1 checkpoint by molecules like P53. Imagine if any cell is detected during this G1 phase that has not fulfilled any one of these requirements, then it will not progress into this S phase. Like in the same way, at the G2 checkpoint, they check the cell size and whether the each cells have replicated and each chromosomes have replicated evenly, or whether there are enough protein reservoirs within the cell. They check for these requirements. If those cells, if any cell which do not require any of these requirement, if any of those cell does not fulfill any of these requirement, they will not go into this M phase. Then and there the cell cycle stops and they divert those cells into repairing process. Here then we will further discuss about P53 molecule. I told you earlier there is a molecule called P53 which detect those abnormalities during G1 checkpoint, and here we going to discuss about this P53 molecule. Actually it is a tumor suppressor gene. P53 is a tumor suppressor gene, and also it is called as guardian of the genome. It has given the name guardian of the genome because if a cell undergoes cell cycle process, it can detect any abnormality and it protects the cells getting abnormal and thereby proliferating and making tumors. So tumors are prevented, tumor formation is prevented by this P53 gene. So now you can understand P53 is very important for a normal human being. If there is a patient with any carcinoma, you can understand this P53 gene has been mutated, or its function is disrupted. or it cannot done its function properly. When there is DNA damage or oncogenic stress or hypoxia occur, this p53 molecule activate. This p53 molecule activates by phosphorylation. There are three mechanisms that can be done by p53 gene. There are three mechanisms which can be done by p53 gene. They are activation of temporary cell cycle arrest, that means quiescence, induction of permanent cell cycle arrest, that means senescence, triggering of programmed cell death, that means apoptosis. There is another gene called RB gene or retinoblastoma protein gene. RB gene or retinoblastoma protein gene. It acts as a sensor of external signals that come from the mutation. Then p53 can act as a central monitor of internal stresses. And when it detects any of the external signal or mutation of the cell, p53 diverts those stressed cells into one of these pathways. First, it diverts this first pathway or temporary cell arrest. Thereby, it gives time for the cell to repair in process. If it is cannot repair, then the cell undergo one of these two processes. That means the cell undergo permanent cell cycle arrest or programmed cell death, that means apoptosis. If we consider the first pathway or quiescence or activation of temporary cell cycle arrest, it is considered as the primordial response to DNA damage, and this occurs in late in G1 phase. After the activation of p53, there is p53 dependent transcription of cDKNIA gene, or we call it as p21 gene. Here you can see this gene is activated. Then p21 gene do inhibit cyclin CDK complexes. It acts as a CDK inhibitor, so it inhibits cyclin CDK complexes and prevent phosphorylation of RB gene, thereby arresting the cell in G1 phase. When the cell undergo G1 phase or arresting in the G1 phase, that means this gives the cells time to repair DNA damage. If there is successful repair occur, then again p53 upregulate transcription of MDM2 and it relieve the cell cycle block. That means it relieve the cell from the block and it allows to continue the cell cycle of that cell. Imagine if this cell cannot successfully repaired, then that cell may enter into p53 induced senescence phase or apoptosis phase. So the senescence or induction of permanent cell cycle arrest phase requires activation of p53 and ORB genes and expression of their mediators such as CDKI. You don't have to remember such details. And in the third one, apoptosis, this is the ultimate protective mechanism against neoplastic transformation. It is mediated by upregulation of several pro-apoptotic genes. So, those who have a defect in the p53 gene or mutation in the TP53 which encodes p53 gene, there is a syndrome called Li-Fraumeni syndrome. So that's all for today. Rest of the details will be discussed from my next video. Thank you for watching.